This video is about abstract on triploid strip catfish, Pangasianodon hypothalamus, growth performance and gonadal development journals. These journals are by Yusran Ibrahim, Tina Trisoy Dorothy and Ardan Carmen. The aim to evaluate from this journal is the growth performance and development of diploid and triploid on strip catfish, Pangasianodon hypothalamus. Before treatment were done, triploid and diploid were separated through nucleolus counting. Triploid were produced by heat shock method at 42 Celsius for 2 minutes at 2 minutes after fertilization. The sample used were 5 individuals each for triploid and diploid at age of 9 months, and the sample were tagged and period for 2 months. The parameters analyzed were growth performance, feed conversion ratio and survival rate using independent sample t-test at confidence interval 95% and gonadosomatic index GSI and gonad histology. What can we observe is there are no significant differences were observed between diploid and triploid fish in terms of growth, performance, feed conversion ratio and survival rate, while the GSI was higher in diploid compared to triploid females. The histological observation of triploid for female is the gonad shows early development stage indicating sterility. The symptoms are oocyte devolution and abdominal development, while male gonads develop faster compared to female as spermatids were found in several lobules. As the conclusion, diploid and triploid strip catfish Pangasianodon hypothalamus growth performance did not differ up to the age of 11 months, although some sterility symptoms were observed in both male and female.
Hi everyone, Pangasianodon hypothalamus is a strip catfish which is a high economic value community has been widely produced in Indonesia for many years. According to Fisheries Republic of Indonesia 2016, its price range between 6 ringgit Malaysia to 8 ringgit Malaysia per kilo for consumption size. The strip catfish fillet industry continued to grow as the demand increased on both local and international markets. However, in the last few years, strip catfish production has been marked by some efficiency problems such as high operation costs, which do not balance with the price on the market. This is mainly due to the slow growth performance of the strip catfish. Triploidization is defined as a part of the chromosome set engineering method, which transforms a 2-set chromosome diploid into a 3-set chromosome triploid. It produces sterile fish with no gonadal development, resulting in a control of wild population, survival rate, growth performance, and meat production increasement. Nowadays, triploid fish are widely farmed in Europe because of their ability to overcome various problems, such as early gonadal maturity, which may lead to premature harvest of the fish. And it also can prevent genetic interaction between farm and wild fish. Theoretically, triploid fish show a better growth performance compared to the diploid one, especially in adult fish, which use the nurture for somatic growth rather than gonad maturation. However, based on Taylor 2014, the mentioned theory was not proved for some species such as Atlantic salmon and Atlantic cod, which show a lower growth performance of triploid compared to diploid. Triploid fish can be produced by inhibiting the release of the polar body 2 during the second stage of meiosis, which occurs in some species. When the zygote reaches 3 to 7 minutes of age, triploidization can also be done by physical, like heat shock, the shock of room temperature, on the pressure, and chemical by using anti-clouding substances such as colchicine, vitritin, and tetrahydrocin. Thus, this study was aimed to evaluate the growth performance and gonadal development of salmon, diploid, and triploid strip catfish up to 11 months of age.
Methodology. Part A. We will discuss about spawning, artificial fertilization, and triploid induction. Throughout this experiment, 3 matured Siamese striped catfish females and 2 matured males were used. Average body weight of the females was about 2 to 3 kg with age of 2 to 3 years. Meanwhile, the male weighed 1.5 to 2 kg and age 1.5 to 2 years old. Spawning was artificially done by using induced breeding technique. The females were induced with HCG hormone 500 IU per kilogram body weight. After a period of 24 hours, the females were induced again with ovaprim at a dose of 0.6 ml per kilogram of body weight. Meanwhile, males were also induced with ovaprim with dosage 0.3 ml per kilogram of body weight, and after 8 hours, they were stripped. Eggs were washed with a soil suspension after the fertilization process to avoid adhesion. Triploid induction was carried out at the age of 2 minutes after fertilization through heat shock with 42 degrees Celsius for 2 minutes periods. Eggs were finally incubated at 28 to 29 degrees Celsius. Fertilization rate was determined 6 hours after incubation, while hatching rate was determined 2 hours after the eggs hatch. Next, Part B. Triploid identification. Diploid and triploid identification was done by calculating the nucleolus. Ploidy level confirmation was done on 2 weeks old fish. Lastly, Part C. Fish rearing. Before rearing the fish, they were screened to separate diploid and triploid fish. Then, the fish kept separately in floating nets in order to determine growth performance and gonadal development. Fish were tagged using a passive integrated transponder, also known as bit tag. Fish were fed twice at satiation. Data on growth performance and survival rate were collected weekly, while feed conversion ratio, or FCR, was determined every 2 months. Gonad observations were carried out on 10 months old fish.
Hello, this time I gonna tell you how about the result of this research. Let I show you through this video. Based on the table show the result of fertilizing rate and hatching rate, in comparison for both diploid and triploid treatment. From the table researcher described there is no significant difference in fertilizing rate. However, there is significant difference in hatching rate between diploid and triploid treatment. Figure 2 show the chromosome and nucleoli analysis by microscopic observation under histological stain. The reported results show triploid have 81 chromosomes higher than diploid, that is 54 chromosomes. Furthermore, the maximum number of nucleoli in triploid is 3 nucleoli and diploid is 2 nucleoli. Based on Table 1 shown the growth performance analysis data. From that, we can say the growth performance between diploid and triploid striped catfish is not significant different. It is because the percentage of variation between growth performance in each treatment is not too diverse. However, the growth rate in triploid is slightly higher than diploid but not significant. Based on Table 2 shown the gonadosomatic index GSI. The GSI for both male and female triploid is smaller than triploid. So, it is indicative the disordering gonadal development of triploid catfish. Based on theory, triploid will use sparing energy for somatic growth and do not use it for vitellogenesis. Figure 3 described that diploid gonad is bigger than triploid gonad. Through histological analysis have shown the abnormalities of triploid gonad. For example, triploid female gonads show the oocyte degradation and only small proportion reach the pre-vitellogenic phase. Beside, triploid males show many empty space between cell.
For this question, based on the result, it is shown that eggs are harvest smaller as well as the success rate of triploidization greatly depends on the temperature. Egg quality also play an important part in the triploidization success, where the low hatching rate observed in triploid is a consequence of the heat shock, and it is to reboot the embryogenesis process. The shock treatment can generate a mesh to the microtubule and improve intratriploid fish growth performance depend on the gonad development activity on the fish. Gonad histology demonstrates a fast gonadal development in male fish compared to female, and diploid male gonad reach the phase of spermatic, and triploid male gonad have the ability of producing sperm.
Conclusion. Growth performance of diploid and triploid Pangasianodon hypothalamus did not differ up to the age of 11 months. Testes and ovaries of the triploid fish showed abnormal symptoms, which did not cause permanent sterility.